Good morning. As always, empty room, locked door, so I'm by myself. Biology 1300, Chapter 8, How Cells Reproduce. I have not turned this thing on, so it's not going to work. Hang on just a second. I apologize. All right, so Henrietta Lacks, an African-American lady who died of cervical cancer more than 50 years ago. But her cells still live on in research laboratories. When her tumor was removed, they took some samples of it. Those cancer cells started to reproduce, and they are still reproducing to this day. They are used widely to investigate cancers, viruses, the synthesis of proteins, the effects of radiation, basically anything that could be tested on cells in a laboratory. The HeLa cells have been involved in it. If you've taken a vaccine, medication, any other kind of drug treatment, you should say, thank you, Mrs. Lacks, for your cells. One thing we have to understand about cancer cells is that cancer cells are considered immortal. Now, some of them will stop growing when they're outside of a body, but they've lost the gene, which is called a terminal gene, that signals for that cell to stop replicating. Cancer cells have lost that, and they will keep replicating as long as they are nourished. We are not immortal, we all know that. And to understand this, we have to look at the structures and mechanisms that cells use to divide. Here's a picture of Miss Lacks, and on the right-hand side is a picture of a pair of her cells. This is the two of her cells in the process of dividing. The mutation is occurring right in here. If you look at the slides individually in your on your textbook site, you will see that below these slides there's a little note explaining what's going on right here. If you want to do that, it's fine. I'm not going to ask you any questions about that. We need to know about the HeLa cells. So the way cells divide is through multiplication. The cell must first multiply, and then it can divide. So a cell reproduces by dividing in two. Each descendant cell receives a full set of chromosomes and some of the cytoplasm. But before the cell can divide, it must duplicate or multiply its genetic material, or the chromosomes. The process cells use to do this is called mitosis. It is nuclear division that maintains the chromosome number in the daughter cells. The original cell is referred to as the mother cell. The two new cells are referred to as daughter cells. This type of division is used in growth, in development, replace damaged cells. Our red blood cells, for instance, only have a limited lifespan. We have to make new ones all the time. And the way new red blood cells are made is through the process of mitosis. When you're fully grown, you still have to replace cells on occasion. Skin cells, for instance, have again a finite lifespan, and they have to be replaced. The process by which they are replaced is through mitosis. Also, organisms that undergo what is referred to as asexual reproduction use a type of mitosis to grow, and to reproduce that way. The cell cycle is a three-stage set, and actually it's more than that, but again, it's a series of events from the time a cell forms until the cytoplasm divides. The cell cycle is usually broken into two parts. You have the mitotic phase, which is this area right here, and we'll talk in some pretty good detail about that in just a moment. The other part is called interphase. Interphase is when the cell is doing all of its normal living and growing and processing and making proteins and all those things that cell has to do. But again, three phases. You have the interphase, which is all of this, steps 1 through 4. You have the mitotic phase or mitosis, which is steps 5 and 6. And then the third phase is cytoplasmic division. Sometimes you will see the word cytokinesis written instead of cytoplasmic division. Most of the cell's activities occur during interphase. Again, interphase is divided into 3 stages as well. But in interphase, the eukaryotic cells, this is where, again, all of the life phases of that cell are going on. The cell produces the proteins it needs to do. It does the cellular respiration, which we talked about in the last chapter. All of the daily processes that have to happen by that cell are done during interphase. The first stage is called G1, which the G stands for gap. Some researchers, some authors will use the word growth phase 1, but a better term is gap. This is the interval between of growth before DNA replication. The second stage is the S phase, which stands for synthesis. It is during the synthesis stage that the DNA for that original cell is being replicated. We have growth and development during G1. We have the synthesis of the new DNA in the S phase or stage. And then the second gap when the cell prepares to divide is G2. Human beings have 23 chromosome pairs. We talked about that in a previous chapter. Except for a pairing of sex chromosomes, which is X and Y in males. The chromosomes of each pair are homologous. Now, this term homologous means they are the same. Again, we have to be real careful when we say that these homologous pairs. Each one has carries the same genes for the same trait. The specific information may be different. And what I mean by that is there is a spot on each gene that is responsible for, let's say, hair color. All of those chromosomes are going to have that same hair color gene in the same place. One person may have a gene for blonde hair, and another person for black hair, and another person for brown hair. So hair color is what is referred to as the phenotype. But the gene, the genotype, is always there. It's in the same place for everybody. Homologous chromosomes are members of a pair of chromosomes with the same length, shape, and genes. One member of that pair was inherited from your female parent, the other from the male parent. And again, this is one of those things that can get kind of confusing when we start talking about how plants reproduce. If we get that far in this course later on, you will find out that plants actually do have male and female components, just like animals do. Again, the eukaryotic cell cycle, phase number 1 shows the cell spends most of its life in interphase, which includes those 3 stages, and we've already mentioned that. Let's drop all the way down to number 5. The nucleus divides during mitosis. The 4 stages are detailed on the next few slides. The cycle will begin anew once this cytoplasmic division, again, that's sometimes called cytokinesis, depending on the textbook or the authority you're looking at. And then all along the stage, there are built-in checkpoints to make sure things are happening the way they are supposed to happen. So we start off with a cell in interphase. On the left-hand side, we have a typical cell, just a sort of an artist's rendering of the cell. You see a pair of centrioles will start to form in the cytoplasm. We still have a well-formed nucleus here. But we talk about plant cells and animal cells, and we'll show the difference in those with these slides. But this is interphase. The pair of centrioles will begin to form, and then the cell will enter what is called early prophase. Again, this still is a nucleus. You still see a very well-defined nucleus in both of those. But you can see some structures are starting to begin to form in here that weren't there before. And you don't see those things. All you see is just these 2 big sort of nucleoluses. And there's a big nucleus. Now you can start to see some little structures starting to form. Again, here's the artist's rendering showing the chromosome pairs starting to coalesce and coil together and then the different member of each homologous pair comes together. Then during prophase, we now have fairly well defined chromosomes, and again, they're not very visible, very well visible here or here. You can see them a little better in the animal cell than you can the plant cell. These little lines are the chromosomes that are starting to pair up. Microtubules begin to form, and there's those centrioles that were forming on the outside. They've now sort of oriented themselves on opposite sides, and the microtubules will form, and they will grow from one centriole to the other, and then each one of these pairs of chromosomes will attach itself to one of those microtubules. During metaphase, all of the chromosomes, which you can now see are well defined, they're all sort of hanging together here, so you can't see the individuals, but they line up in the center along those microtubules. This is metaphase. At metaphase, all of the chromosomes are aligned in the center. The next step is anaphase. At anaphase, the microtubule breaks down, and as it does, it's going to pull this pair of homologous chromosomes apart, where one's going to go over here and one's going to go over here. And you see where you've got the microtubules pulling the different structures. Again, now much better seen in the onion cell than in the animal cell. But you can still see there's one group of homologous chromosomes, and there's the other group. And they're being pulled to opposite sides. During telophase, or again, some people pronounce it telophase. I've always said telophase because I do not answer a telephone. I answer a telephone, so I say telophase. But here you can see, again, in the artist rendering, you have two cells starting to form. You have this area right here, which is called a cleavage furrow, or a cleavage furrow. It's also called a constriction ring can form here. Starts to pinch the cytoplasm in two, but now you see you've got one short red, one short red, one short blue, one short blue, long red, long blue, long blue. So the chromosomes have now divided and separated. If you go all the way back to the beginning, when you don't see anything, the first thing we see is chromosome. You see two reds, two blues, two reds, two blues. After telophase, again, you see the constriction ring or the cleavage furrow start to form here. In plant cells, there's another step that has to happen, and we'll talk about that in just a moment, because plant cells are not only divided by a plasma membrane or a cell membrane, they're also divided by a cell wall. During mitosis, the complete set of chromosomes is distributed into two different nuclei. During G2, each chromosome consists of a replicated DNA molecule attached at the centromere. When these sister chromatids are pulled apart, each becomes an individual chromosome in a new nucleus, and the cytoplasm divides, and the two nuclei are packaged into two separate cells. And again, just a simple artist drawing here. Here's my original cell with two chromosomes. Here's my cell. By the end of G2, we have two chromosomes, and then here's the separation. You have two new cells that look just like our original cell. The cell cytoplasm divides between late anaphase and the end of telophase. We wind up with two cells, each having its own nucleus. And the mechanisms by which the cytoplasm divides differs between plants and animals. It's a much simpler process in animal cells than it is in plant cells. In animal cells, a contractile ring, or as another textbook called it, a constriction ring, pinches the cytoplasm in two. That contractile ring contains microfilaments, which contract when its component proteins are energized by ATP. There's ATP again. Remember, we talked about how important it would be in a previous chapter. Then a cleavage furrow forms. It deepens until the cytoplasm is pinched in two. Each new cell then has its own nucleus, its own cytoplasm, and is enclosed by its own plasma membrane. And again, the artist's rendering of here's the original Here's the cleavage furrow starting to form. The contractile ring pinches even more, and ultimately you wind up with two individual cells. In plant cells, however, it's a little bit more complicated because in addition to forming separating and forming individual cells surrounded by a plasma membrane, we have to have individual cells surrounded by a cell wall, or separated rather by a cell wall. So the microtubules guide vesicles from the Golgi body and the cell surface to the division plane. It's going to be the area where the two cells are going to divide. Those vesicles will contain the structural components needed to build a new cell wall. They will arrive at the structure called the disc plate. This plate grows, forms a new cross wall, the up and down part between two walls. And finally, the cell plate develops into two new cell walls, separating the two dependent cells. And here's that structure. Here's all the chromosomes being pulled apart. Here are all those vesicles that have come with their wall-building components. Those vesicles start to break down and form the new wall, and eventually you wind up with two cells separated by a cell wall on either side. Now, we started the chapter talking about the HeLa cells and their role in cancer. When a cell divides, and when it doesn't, is determined by gene expression controls. What happens in cancer cells is those genes, called checkpoint genes, are disrupted. They don't function properly. And in the case of cancer, what they do is they make the cell divide too quickly. A cell that should be dividing only maybe every 7 to 10 days is dividing 4 or 5 times a day, is one of the things that can cause cancer. These checkpoint genes can become disrupted. Many times that disruption can be corrected after only 1 or 2 generations of the new cell. The disruption will be corrected. The body of a multi-celled organism will recognize that that cell is inaccurate and will eliminate that line of cells through other means. But if that problem remains uncorrected, other checkpoint proteins may cause the cell to self-destruct. And in most cases, that's what happens. We have every time our cells, well, not every time, but lots of times when our cells reproduce, their errors are made. But most of those errors, as we talked about in a previous chapter when we talked about mutations, most of those errors are corrected. Occasionally, those errors are not corrected, at which point can lead to problems. The first thing that can go wrong is a group of cells can start reproducing inaccurately, and that inaccuracy can start to build up, and you can form what is called a neoplasm. A neoplasm is nothing more than an accumulation of abnormally dividing cells, and again, in most cases, this is going to be limited to a very few numbers of cells. Those cells are going to be, again, as said here, some specific proteins will cause those cells to self-destruct, but there is the possibility that those can build up enough to where you start to have a visible or a palpable collection of cells. If the neoplasm continues on to the point where we can see it or feel it, that is referred to as a tumor. Tumor is a neoplasm that forms a lump. This is something we can feel or see. Any gene that can transform a normal gene into a tumor cell is referred to as an oncogene. These are genes that have proteins that promote mitosis, genes that promote mitosis are called proto-oncogenes, and mutations in those can turn them into oncogenes. Again, just think about onco is going to be the tumor-causing genes. Genes that code for receptors for growth factors can be manipulated or can be disrupted, and that can lead to, again, much faster than normal production of new cells, which can lead to neoplasms forming, then to tumors forming, and then you get the tumor that you can see or feel. Checkpoint genes also are the products of checkpoint genes that inhibit mitosis are called tumor suppressors because tumors form when they are missing. An example of tumor suppressor genes are products of the BRCA1 and BRCA2 genes. Those are genes that are responsible for BR, breast, CA cancer. These are genes associated with breast cancer. Viruses such as HPV, the human papillomavirus, cause a cell to make proteins that interfere with its own tumor suppressors. You see a lot of commercials on television now, the ones with the kid. You'll start with the young boy or girl when they're 7 or 8, and they're asking their parents, did they know? And then the kid is 10, and then the 12 is 16, and then it's the kid is 19, and the kid has developed some kind of cancer. The HPV virus, the human papillomavirus, it's one that almost all of us have been exposed to. Virtually every human being on the planet has been exposed to the human papillomavirus. Most of us, our immune system has the ability that we clear it. It's one that doesn't stay in our system forever. It's one that we get and we clear it. We get rid of it. And then the next time we get exposed to it, we get rid of it again. The vaccine for HPV simply enhances that natural ability for your immune system to do its job. But what can happen is the human papillomavirus is the one that causes warts and some other things, can actually cause cancers later in life. And that's what these commercials are saying. Doctors are recommending that all pre-adolescents have a human papillomavirus. Again, my wife's not a practicing nurse anymore. She's retired. But if she was still practicing, I could just call her on the phone right quick and say, hey, what is the age at which you're supposed to, and I think it's you're supposed to have it by the time they're 12 or 13. But again, check with your pediatrician about that. But again, we don't want this virus to disrupt its own cell. Benign neoplasm, such as a mole, is non-cancerous. A malignant neoplasm is dangerous to your health. Again, lots of different benign tumors. Again, when we hear somebody say they have a tumor, we're usually talking about some cancer issue. Most tumors are actually benign. If you look at the total number of tumors that human beings have, if you've ever watched Dr. Pimple Popper and she's squeezing out all those lipomas and all those other things, well, those are tumors. But most of them are benign. So most tumors are benign. Cancer occurs when the abnormally dividing cells of a malignant neoplasm disrupt body tissues physically and metabolically. In other words, when the cancer cells or the tumor cells start to invade organs or push organs out of the way or disrupt them the way they function, then that is what causes cancer, or what is labeled as cancer. Malignant neoplasms can break free and invade other tissues. That is called metastasis. You've heard where someone had cancer and it had metastasized. They had lung cancer, but it had metastasized to their brain. That simply means that the lung tissue that was cancerous broke free, flowed through the bloodstream, and then lodged in the brain to form brain cancer. Drawing here's a neoplasm. It's benign. It grows slowly, stays in their home tissue. They don't move. Here's the cells of the malignant neoplasm. You see the cells, but you see the cells are starting to break free and wander. They can become attached to the wall of a lymph vessel or a blood vessel. So blood vessel is shown here. They can release digestive enzymes that create an opening in the wall there. Enter the vessel. Now they can flow through the bloodstream, where they can then do the same thing again and creep into other tissues. Just like I mentioned, you can have, back up. This is lung tissue, for instance. This cell broke loose, is now going through the bloodstream, and now, boom, it's in your liver. So it was in your lung, now it's in your liver. Again, cancer cells grow and divide abnormally. You can remember two things about cancer cells. They generally reproduce faster than normal. So again, that cell that normally should be dividing every, I don't know, 7 to 10 days is dividing 3 or 4 times a day. So they're abnormally dividing, and, or excuse me, their growth is abnormal, and they don't make really good copies. Keep in mind, every cell in your body has the blueprint to make a whole new copy of you. But skin cells make skin cells, and liver cells make liver cells, and kidney cells make kidney cells. But occasionally in these tumors, this is really kind of interesting, and this will actually occur in these types of tumors as well. But the doctor will go in, he says, oh, this is a tumor, or this is a tumor, this is a tumor I can remove, right? So I'm going to remove the entire tumor. The tumor is removed. It is then sent to the pathology lab so they can check it to see if it is in fact a cancerous tumor or a benign tumor, if it's malignant or benign. They will then start to take that tumor and they will slice it to take thin slices so they can look at it microscopically. And it is not unusual at all that deep inside these tumors to find things like hair and teeth. And you go, hair and teeth? Yes. Because the cell was disrupted, it was not using the proper part of the DNA, and it actually started using the DNA that's used to make teeth or used to make hair, and those things would form. Now, they wouldn't look exactly like a tooth that you would find in your jaw, but they are teeth. They are structurally teeth, structurally hair, found inside of these cancer cell tumors. They may have an abnormal plasma membrane, they may have an abnormal cytoskeleton, they may have an abnormal chromosome number, and their metabolism. Again, generally metabolism is going to be sped up in these cells. Cancer cells don't stay anchored properly in the tissues because the plasma membranes for adhesion are defective or missing, so these things can wander around the body, whereas the benign tumors don't do that. There are pieces of non-decoding DNA sequences at the end of eukaryotic chromosomes called telomeres. These provide a buffer against loss of more valuable DNA. Basically, if during all of these processes of mitosis, meiosis, et cetera, if something goes wrong and a piece of the chromosome breaks off, it's more than likely going to be the tips or the ends. And by having these telomere present, if the very tip end of a chromosome breaks off, that's information that's not really needed. It's not vital information, so we don't lose, the cell doesn't lose any vital information. Under normal circumstances, eukaryotic chromosomes are shortened by about 100 nucleotides for every replication cycle. If the telomeres are too short, it can trigger a checkpoint that will halt the cell cycle, and this may play a role in setting an organism's lifespan. Why do some things live longer than others? Human beings have changed over the past several hundred years. In the 13 and 1400s, if you lived to be 50 or 55, you could consider yourself having lived a long and healthy life. Now, if you die at 55, well, that guy was just a child. He was a youngster. That's way too young to be dying. Well, lots of things can play a role in that. Basically, most researchers believe that human beings are just now getting to what should be our lifespan. As a matter of fact, there are some researchers that have suggested that the actual normal human lifespan should be about 100 years, a little more than 100 years. Why do most people not live that long? Because of outside influences that are messing with our cellular lifespan. Paired genes on homologous chromosomes can offer a variety of, or can vary slightly in DNA sequences. Again, you've got the paired genes, you've got the red hair on one side, the brown hair on the other side. Alleles are different forms of a gene that encode for slightly different versions of a gene product. So you have a red hair allele and a brown hair allele. Those are different forms of the same gene. Offspring of sexual reproducers, like you and I, inherit new combinations of alleles, which is the basis for new combinations of traits. Historically, and I'm talking go back 300 or 400 years, if a pair of brown-eyed, brown-haired people made a baby and the baby was born with blonde hair and blue-eyed, the woman would be taken to the county or to the government seat and either hung or stoned to death because she had clearly cheated on her husband, and that baby was not her husband's baby because he had brown hair and brown eyes and she had brown hair and brown eyes. How did we wind up with a baby with blonde hair and blue eyes? Again, we didn't understand these processes. Alleles, again, showing this is an actual set of chromosomes that have been colored to show the different genes. Here we've got an artist's rendering of the things, and you see genes occur in pairs on homologous chromosomes. So this is a gene, and this is a gene. This is a gene, and this is a gene, et cetera, et cetera. And you'll notice it says right here, color variation represents sequence differences. So you got that one is a slightly more yellow than that one. To my eye, that one's a darker color than that one is. This is darker than this one. These two appear to be about the same color, basically saying the information could be the same on either side, but the information could vary from one side to another. What are the advantages of sex? Well, both sexual and asexual reproduction have their advantages and disadvantages. Asexual reproduction, offspring arise from one parent. Offsprings are genetic clones of their parent, and therefore there is little genetic diversity. That's a disadvantage. The advantage of asexual reproduction is you can reproduce whenever you need to. You don't have to find a partner. If you decide, oh, it's time for me to produce a new generation, asexually, you can just do it. The problem is they're all going to be genetic clones. There's going to be no genetic variation. Sexual reproduction, on the other hand, offspring inherit genes from two parents. Diversity offers a better chance of surviving. If you have an environmental challenge, harmful mutation, virus, all of those things, diversity increases the ability to survive. COVID-19, why are some people getting it? And you know, there have been 40-year-olds that get this virus. They start to cough, they get a little worse, they go into the hospital, they get put on a ventilator, and they die. Other 40-year-olds are testing positive, which means they've had it, but they've never known it. They never had a fever. They never had never lost their sense of smell or taste. They had never had a cough. I'm coughing. The reason I'm coughing right now is because my throat is dry. But it's diversity. It's the different immune systems in people. There's the reason that some people are getting it and having a really, really hard time. Other people, not so much. And if you do things sexually, if there is something that gives you a benefit, a mutation occurs that is beneficial to the species, it will spread rather quickly. If you're doing asexual reproduction, what has to happen for a mutation to move from one individual's line to the next? That means every time we need that, every time that organism gets that that benefit, it has to be because of a new mutation. Whereas in sexual reproduction, if I had some benefit, I don't know, whatever, and I passed it on to my children, then they passed it on to their children, then they passed it on to their children. The gene only had to mutate once in me, and now it's spreading throughout the population. Up here, to spread throughout the population, it would have to continually, that same mutation would have to happen. The process by which cellular reproduction works is called meiosis. Now, meiosis is similar to mitosis. It's not exactly the same. It is similar. But what it does is very important. It halves the parental chromosome number by sorting the chromosomes into two new nuclei, and it does it twice. So you separate once, and then each one of those new cells will separate again. This is the basis of cellular reproduction. This is where we get eggs and sperm through this process. Gives rise to new combinations of alleles and offspring. My dad had 2 genes, my mom had 2 genes, but I got one from my dad, one from my mom. That's a new combination. So that's how we get new combinations of alleles. Duplicated chromosomes of a diploid nucleus, 2N, are distributed into 4 haploid nuclei, N. For human beings, the N number is 23. Here are some drawings of mitosis. Now, you're going to see some similar words here. Here's prophase, here's metaphase, anaphase. Now, remember, the interphase stage of these cells is the same. You got G1, S, and G2. But when it comes to the mitotic component of meiosis, or the separation stage, you have prophase 1. In prophase 1, what happens, instead of this red X and the blue X that's under it acting independently, they're going to work together for a while. This is the red one is the chromosome. This is the chromosome you got from your mother. The blue one here is the chromosome you got from your dad. So the red ones pair up, the blue ones pair up, and then anaphase starts. You see the spindle fibers starting to form. There's the centromeres. Now the spindle fibers are forming. And in metaphase, what happens here, instead of all of these things lining up individually, like they did in mitosis, in meiosis, you see they're lining up in pairs. And let's not worry about this right here for just a minute. The fact that this one's got a red end and this one's got a blue end. We'll talk about what that is in just a moment. But this is my metaphase 1. And in anaphase 1, both of these are going to go this way, both of the red ones this way. And so they're separating as a pair. And then 2 cells are going to form in telophase 1. So you've got both red ones and both the long blue ones, and then you got the long red and the short blue. And you see it's forming. These are actual pictures, actual photomicrographs of what's going on. Then these are going to, this happens very quickly. Prophase 2 happens very, very quickly. Basically, all that's going to happen is this new membrane forms. We form 2 fully functional cells, and then they immediately move into metaphase 2. Now, if you'll notice in metaphase 2, we're back to being lined up like we were in meiosis. So if I ask you which stage of meiosis most closely resembles metaphase of mitosis, metaphase 2 of meiosis. Then we have anaphase 2, the cells start to separate. Then we have telophase and the cytokinesis step. And we've now separated into 1, 2, 3, 4 cells. But if we go back to the beginning, we had 1 red, 1 blue, 1 short red, short blue, long red, long blue. Excuse me. And then the duplication. And then over here, we've got 1 short one, 1 short one, 1 short one, 1 short one, long blue, long red, long red. So we've gone from 1, 2, 3, 4, even though that's a duplicated pair, but that duplication has occurred. So the original cell had 1 long blue, 1 long red, 1 short blue, and the last one has 2, so you get to 2. These would be examples of either eggs or sperm. Meiosis shuffles the parental combinations of alleles, introducing variations. You can have something called crossing over, which happens in prophase 1, or you can have random assortment, which happens in metaphase 1. Crossing over is a recombination between non-sister chromatids of homologous chromatids, which produce new combinations of parental alleles. Okay, non-sister, homologous. You have 23 chromosomes. And let's talk about chromosome 5. You have a chromosome 5 that you got from your mom, and you have a chromosome 5 that you got from your dad. The homologous pair of chromosomes would be chromosome 5 from mom, chromosome 5 from dad. That's the homologous pair. When this one duplicates, chromosome 5 from mom, when they duplicate, each one of them is referred to as a sister. So you have two sister chromatids of mom, two sister chromatids of dad. And what will happen or can happen in prophase 1 is that a tail of one of those chromatids from mom and one of those chromatids from dad can get twisted together, and pieces of them can actually change place. That's the recombination between non-sister chromatids of homologous chromosomes. So here's crossing over. This is chromosome whatever. Here we focus on only 2 of the many genes per chromosome. In this example, one gene has alleles A, and another one has A and B. Okay, you see the capital A, capital B, little a, little b. So maybe this is from mom. Let's say blues are colored for boys traditionally. So dad, mom, and then they duplicate. When they come together at prophase 1 and they're stuck together, see here, the crossing over is occurring. You now have the short blue end here. So now you have one of my chromosomes is just like the original, big A, big B. One of them, little a, little b. But what do we have here? We have big A with little b, and over here we have little a with a big B, or uppercase B. That's crossing over. A piece of this chromosome and a piece of this chromosome have changed places. But keep in mind, crossing over always occurs in homologous chromosomes. So it's always a piece of chromosome 5 and a piece of chromosome 5 changing places, not a piece of chromosome 5 and a piece of chromosome 9, for instance. Apologies, I stepped over and got a drink, so maybe my throat will stop sounding like I gargle with gravel. Sexual reproduction involves the fusion of reproductive cells called gametes. Again, I've heard this word pronounced gamete. I was taught gamete from 2 parents. Whoops. All the gametes are haploid. In other words, they have half the number of chromosomes. So again, the 2N number for human beings is 46. So each one, egg and sperm, will have 23 chromosomes. That's the word haploid. They arise by division of immature reproductive cells called germ cells. And at fertilization, 2 haploid gametes fuse and produce a diploid zygote. One of the last words in the dictionary is zygote, which is the first cell of a new individual. Lastly, for this chapter, some points to ponder. What are some ways that cell division and fertilization improves genetic diversity with each generation? Again, each generation will get beneficial things from the previous generation. Why is it difficult to design a drug that will stop cell division only in tumor cells? Because if it stops it in tumor cells, it may stop it in other cells as well, because those tumor cells and normal cells have some similarities. There actually is now some research being done into virus therapy, where they are engineering viruses to attack only very specific types of cells. It's in the early stages. They're doing some of this work over at UAMS in Little Rock. But again, it's in its very early age, very early stages. And we have chemotherapy, radiation, some of the same side effects you see with those, you would see in things that stop cell division. Questions, please contact me at either trussell@uaptc.edu. Exam number 2 should be showing up in Blackboard in just a few days. I'll check to make sure when that is and let you know via an announcement. If you would rather contact me through the course contacts, course messenger link, that's fine too. Have a great day. It's Wednesday where I am, so have a nice weekend if you've got a weekend coming up, and I will see you next time.